Okay, so last unit was everything you didn't want to know about cell structure, and everything in this week is going to be everything you didn't want to know about membrane structure. Okay, so we're going to start off by talking about the different cell membrane components, the most important of which is the phospholipid. So again, membranes are important because it's what's separating the inside of the cell from the outside and just allowing it to control that environment. So within the membrane, you have these things here called phospholipids. And so there is two layers of them, and they form this thing called a bilayer. So taking a closer look at the phospholipid structure, this molecule is said to be amphipathic, and that means that it has both hydrophobic and hydrophilic properties. So all hydrophobic means is that it's afraid of water. Hydro means water and phobic means it's afraid of it, so it's going to avoid it. And that some parts of this molecule are hydrophilic, so they're going to be attracted to water. So let's look more at the structure of the phospholipid. So it kind of comes in two main parts. This first part is going to be, we're going to call it the head, and then the second part is going to be the tail. So when we look more closely at the head structure, we see that it has two different parts. One of them is this phosphate here, and then the other part is this glycerol, and together they make this head structure. So these guys, this whole thing, this part is considered hydrophilic. That means it's attracted to water. So what's happening is you can see that this phosphate has some negative charges on those oxygens, and those negative charges are attracted to the partially charged hydrogens on water. So it's going to mix really well. They're going to form like bonds, and they're going to be attracted to each other. So that's why it's called hydrophilic. Now, in contrast, the tails they have not one but two fatty acids. So one of them is saturated, and then the other one is unsaturated, which it means it has this double bond, and they each contain about 18 different carbons. And in contrast to the phosphate head, these tails are water-fearing. So if we look at what, these fatty acids look like, it's just a big chain of carbons with a bunch of hydrogens on it. And as you can see, there's no charges here on the fatty acid to interact with those positive charges on the water. So basically no interaction occurs, and in fact the water prefers to form hydrogen bonds with itself, and it's going to cage those fatty acid molecules away from the water. So they don't mix. You ever hear that saying like oil and water? It's exactly what's occurring here. So to go back to what I'm talking about, saturated versus unsaturated hydrocarbons, so these are two fatty acids, and again, it's mainly a long chain of carbon. When it's saturated, it does not have any hydrogen bonds between any of the carbons. It's all possible hydrogens are attached. So that's what it means to be saturated, because it is saturated with hydrogens. Now, unsaturated, by contrast, does have some hydrogen bonds in it between some of the carbons, so it doesn't have the maximum amount of hydrogens possible. So they're going to have these sort of bent structures when you have those double bonds, and it actually will prevent overly tight stacking. So that's why you're supposed to eat more unsaturated fats, because these are going to have a harder time clogging your arteries. So again, these heads are hydrophilic, and the center is hydrophobic, so when they get together in, a biological environment, they're going to want to get away from the water, these hydrophobic parts, and the hydrophilic parts are going to want to be towards the water. So they form this orientation of a bilayer where the hydrophilic heads are pointing both outside the cell and inside the cell, and the hydrophilic or hydrophobic parts are in between. So we know a lot about how membranes are structured now, and this is an electron micrograph of a membrane right here. But that knowledge has been refined over a long time. So it started back in the 1920s with Gorder and Grendel, and what they did was they isolated membranes from red blood cells, and they knew the thickness of what it should be if it was a single layer of phospholipids, but they realized that it was twice as thick. So they concluded that it had to be not one layer of phospholipids, but two. So this was the first part of the model, but even though they knew that somewhere in that membrane there were proteins, they did not know where they were. And so when electron micrographs came on the scene, when we started to have those electron microscopes available and they were able to take really good pictures of what a cell membrane looked like, they were able to see these two sort of dark lines forming like a railroad track. And so they said, okay, you know what? This is like a sandwich model. Up here, we have protein, and then in between, we're going to have the phospholipids, and then on the bottom, we're going to have another layer of protein. And so to test this model, you're assuming that it's going to have a flat external structure, very even on either side. But that model did not stand for very long because people started taking something called freeze-etch micrographs. And so what they did is they freeze the cells and then they break the membranes apart, and they're going to split at the weakest point, which turned out to be the center. And when they did that and they're looking at these micrographs inside these, layers, they're seeing lots of bumps. The exterior was a little bit smooth, but the interior had the bumpy globs. And what those bumpy globs were proteins that were located and mixed in with the phospholipids. So this basically killed the Davidson-Denali model because this is the membrane of a nucleus. And you can see that this surface is not smooth. It's got a lot of stuff sticking out. We can see some nuclear pores. It means that there's protein embedded inside the membrane, and when we look inside that, you see all these dots too. So there's protein contained inside, not just a flat layer on top. So later, they were able to start extracting membrane proteins and start figuring out their structure. And these are just some examples of like different proteins that you might find in the membrane. And you can see that their structures are really different from each other. Some of them are smaller, some of them are larger, and they have really different shapes. You would expect, if it was the sandwich model, that there would be a single regular shape that was going to go over the entire cell. But that's not what they're finding in there. And this sort of dovetailed with what they were seeing in the freeze-etch micrographs because they're seeing all these bumpy, irregular structures. It was not flat and smooth. And what's more, when they started looking at the amino acid sequences, they found that the parts that were embedded in the membrane had those hydrophobic regions just like the fatty acid tails, which would make sense about why they would be in between instead of on top. Because if they were on top and bottom, you would expect them to be hydrophilic. And since you didn't have if you have those hydrophobic regions, that model cannot be correct. So the other thing that they did was to look at the fluidity of a membrane. So what they did is they take two different cells, and in one of the cells, they take the membrane proteins and they tag them with a green antibody. And then in a different cell, they take the proteins from there and they tag them with a red antibody. And then they fuse together those cells and they wait and they see what happens. And what happened is that the red and the green spots were mixing together. So the membrane proteins did not stay in one place. They were free to move around the membrane, which suggests that now I have this sort of fluid structure in the membrane. It's not static. So along comes Singer and Nicholson, who said, okay, we have this phospholipid bilayer, which is what we're seeing in the freeze etch, and it makes sense because we have this hydrophilic tails that are going to want to be away from the water, and we have the hydrophilic heads, which are going to want to interact with the water, so they're going to be both facing outside and inside the cell. And there's going to be proteins, either on the top, they can be attached and slightly embedded. There's going to be proteins that move through the phospholipid bilayer. And that wherever these proteins are, whether they're on the outside as peripheral or they're spanning the membrane as an integral membrane protein, they have the ability to move around. So this is known as the fluid mosaic model. And it's the one that currently still stands today. That doesn't mean that we're not going to find out anything that would change our perspective there. It just means this is where we are today. So membrane proteins come in two main classes. Integral, which means it's going to span the entire membrane. And because, remember, this is hydrophobic in here, it's kind of oily. The integral proteins are mainly made of hydrophobic amino acids. And we also have these peripheral proteins that are either partially embedded in the membrane and have some region sticking out. So the part that's in the membrane has to be hydrophobic amino acids, and the part outside has to be hydrophilic amino acids. So just like the phospholipids have hydrophilic regions on the outside and hydrophobic regions when it's inside the membrane, the peripheral proteins have to behave the same way. So there are many, many different kinds of membrane proteins. You are only responsible for knowing a few of these different kinds. So they have many different functions. So one of them is a hormone binding or a receptor site of some kind. So these guys are taking signals from outside the cell and passing on the message to all of their friends inside the cell. So in order to do that, part of it has to be outside the cell, which is going to be a hydrophilic region, and it's going to bind to something, some sort of signal, whether it's a hormone or a synapse, whatever. It's going to bind those signals on the outside, and then it's going to send that signal through its transmembrane region to its signaling region in the cell and pass that message on. So those are your receptor binding sites. The other kind of thing that you might have is an enzyme that's embedded inside the membrane, because maybe you want to do some chemical reaction at the membrane, and it can be on either side, inside or outside. But in either case, the active site is going to be exposed to either the exterior or interior of the cell because it has a job. It needs to, catalyze some sort of chemical reaction. So you have those immobilized enzymes. The other thing that you might need is cells need to attach to each other in order to form tissues and organs. And the way they do that is through cell adhesion molecules. So you'll have a cell adhesion molecule in one cell that's going to connect to another one from a neighboring cell, and this allows them to stick together and form those tissues. The other thing you might have are cell-to-cell communications. So these act as cell identifiers. So you would have some sort of, protein, and it's usually a glycoprotein that's sticking outside of the cell. And it's like telling other cells, this is what I am. So an important part of this is for your immune system. All of your cells that are in your body have a special identifier that's on the surface of our cells, so that when an immune cell comes along, it doesn't kill it. It's like, oh, you're identifying a self. They can also act as receptors to transmit signals between other cells. So these would be like the receptors for nerve signals. So the neurotransmitters are going to come out from one cell, be identified on a neighboring cell through these cell-to-cell communicator receptors, and again pass on the signal to that cell. You might also have channel proteins. So this is a kind of passive transport. There's not really a gate. It's just making a space for certain small, non-charged hydrophilic particles to go through that membrane. This is called facilitated diffusion. There is no energy cost here. So it's just an open channel, and it's just letting certain things diffuse through. So it does not cost any kind of energy. This is in contrast to a pump protein, which does burn ATP. This is called active transport because it needs to actively use energy to move the cell, whatever it's moving from outside the cell to inside the cell, and it can go either direction, in or out. So these are your pump proteins. Okay, so now we're going to talk about this special molecule called cholesterol, which you've probably heard something about, a lot of dietary needs that you need to avoid it. So what cholesterol is it is a steroid lipid component of the cell membranes, but you only find it in animals. You will not find it in plants or bacteria. Cholesterol is strictly an animal thing. So cholesterol, because it's mainly these carbons and hydrogens, it is hydrophobic. It's going to want to hide from the water, or more accurately, the water is going to push it around to itself. So you would expect to find it inside the membrane, and in fact you do. You find it kind of near the phosphate heads. It's near the edges, not really in between. And what cholesterol does is it both reduces membrane fluidity and prevents these membranes from being completely solid. So it's keeping it in sort of a happy state of not too much fluidity and not solid. It's helping to maintain that homeostasis of that membrane. And it also helps to block the random permeability of certain solutes. So it's helping to control what is going through here. If it wasn't here, a lot more stuff that shouldn't be getting into the cell would get in. And because of this, if its structure, it helps to give the membranes its curved shape, because most of